Welcome students. Last episode we discussed the transcription in prokaryotes and eukaryotes. In this episode we will discuss the different types of RNA and their functions. Ribonucleic acid is a type of molecule that consists of a long chain of nucleotide units. Each nucleotide consists of a nitrogen base, a ribose sugar and a phosphate. RNA is very similar to DNA but differs in a few important structural details. In the cell, RNA is usually single stranded while DNA is usually double stranded. RNA nucleotides contain ribose while DNA contains deoxyribose. That is a type of ribose that lacks one oxygen atom, and RNA has the base uracil rather than thymine that is present in DNA. RNA is transcribed from DNA by enzyme called RNA polymerase and is generally further processed by other enzyme. RNA is central to the synthesis of proteins. Here, a type of RNA called messenger RNA carries information from DNA to structure called ribosomes. These ribosomes are made from proteins and ribosomal RNAs which come together to form a molecular machine that can read messenger RNAs and translate the information they carry into protein. There are many RNAs with the other roles, in particular regulating which genes are expressed but also as the genome of most viruses. Ribose nucleic acid. Most cellular RNA is single-stranded, although some viruses have double-stranded RNA. The single RNA strand is folded upon itself either entirely or in certain regions. In the folded region, a majority of the bases are complementary and are joined by hydrogen bonds. This helps in the stability of the molecule. In the unfolded region, the bases have no complements. Because of this, RNA does not have the purine pyrimidine equality that is found in DNA. RNA also differ from DNA in having ribose as a sugar instead of a deoxyribose. The common nitrogen bases of RNA are adenine, guanine, cytosine and uracil. Thus, the pyrimidine uracil substitutes thymine of DNA. In regions where purine pyrimidine pairing takes place, adenine pairs with uracil and guanine with cytosine. In addition to the four bases mentioned above, RNA also has some unusual bases. Chemical structure of RNA. An important structural feature of RNA that distinguishes it from DNA is the presence of hydroxyl group at the second position of the ribose sugar. The presence of this functional group causes the helix to adopt the A form geometry rather than the B form most commonly observed in DNA. This results in a very deep narrow major groove and a shallow and wide minor groove. A second consequence of the presence of the 2'-hydroxyl group in that is conformationally flexible regions of an RNA molecule. It can chemically attack the adjacent phosphodiester bond to cleave the backbone. Most cellular RNA molecules are single-stranded. They may from secondary structures such as stem, loop and hairpin. There are more unusual bases in RNA than in DNA. All normal RNA chains either start with adenine or guanine. Three types of cellular RNA have been distinguished. First one is messenger RNA or template RNA. Second one is ribosomal RNA and third one is soluble RNA or transfer RNA. Ribosomal RNA and transfer RNA comprises about 98% of all RNA. All three forms of RNA are made on a DNA template. Transfer RNA and messenger RNA are synthesized on DNA templates of the chromosome, while ribosomal RNA is derived from nucleolar DNA. The three types of RNA are synthesized during different stages in early development. Most of the RNA synthesized during cleavage is mRNA. Synthesis of tRNA occurs at the end or cleavage, and rRNA synthesis begins during gastrulation. First one is ribosomal RNA, that is rRNA. Ribosomal RNA RNA, as the name suggests, it is found in the ribosomes. It comprises about 80% of the total RNA of the cell. The base sequence of ribosomal RNA is complementary to that of the region of DNA where it is synthesized. In eukaryotes, ribosomes are found on the nucleolus. Ribosomal RNA is formed from only a small section of the DNA molecule, and hence there is no definite base relationship between ribosomal RNA and DNA as a whole. Ribosomal RNA consists of a single strand twisted upon itself in some regions. It has helical regions connected by intervening single strand regions. The helical regions may show presence or absence of positive interaction. In the helical region, most of the base pairs are complementary and are joined by hydrogen bonds. In the unfolded single strand regions, the bases have no complements. Ribosomal RNA contains the 4 major RNA bases with a slight degree of methylation and shows difference in the relative proportions of the bases between species. It's molecular with an increasing ionic strength. The molecules show helical regions produced by base pairing between adenine and uracil and guanine and cytosine. Hence, ribosomal RNA does not show purine-pyrimidine equality. The ribosomal RNA strands unfold upon heating and refold upon cooling. Ribosomal RNA is stable for at least 2 generations. The ribosome consists of proteins and RNA. The 70S ribosome of prokaryotes consists of 30 subunit and a 50S subunit. The 30S subunit contains 16S ribosomal RNA, while the 15S subunit contains 23S and 5S ribosomal RNA. The 80S eukaryote ribosome consists of 40S and 60S subunit. In vertebrates, the 40S subunit contains 18S ribosomal RNA, while the 60S subunit contains 28, 29S, 5.8S and 5S ribosomal RNA. In plants and invertebrates, the 40S subunit contains 16 to 18S RNA, while the 60S subunit contains 25S and 58 and 5.8S ribosomal RNA. There are 3 types of ribosomal RNA on the basis of sedimentation and molecular weight. 2 of these classes are high molecular weight RNAs, while the third one is low molecular weight. The 3 classes are high molecular weight ribosomal RNA with a molecular weight of over a million. Example, 21S to 29S RNA. High molecular weight ribosomal RNA with the molecular weight below a million. Example, 12, 18, 188 ribosomal RNA. The third one is low molecular weight ribosomal RNA. Example, 58 ribosomal RNA. The next type is messenger RNA, that is mRNA. Jacob and Monod in 1961 proposed the name messenger RNA for the RNA carrying information for protein synthesis from the DNA to the site of protein formation. It consists of only 3 to 5% of the total cellular RNA. Size of messenger RNA. The molecular weight of an average sized mRNA molecule is about 5 lakh and its sedimentation coefficient is 8s. It should be noted, however, that mRNA varies greatly in length and molecular weight. Since most of protein contain at least 100 amino acid residue, mRNA must have at least 3 times of nucleotides, that is 300 on the basis of triplet code. Structure of messenger RNA. The cell does not contain large quantities of mRNA. This is because mRNA, unlike other RNA, is constantly undergoing breakdown. It is broken down to its constituent ribonucleotides by ribonucleases. Structure of messenger RNA or mRNA. Messenger RNA is always single stranded. It contains mostly the bases adenine, guanine, cytosine and uracil. There are few unusual substituted bases, although there is a certain amount of random coiling in extracted mRNA. There is no base pairing. In fact, base pairing in the mRNA strand destroys its biological activity. Since mRNA is transcribed on DNA, its base sequence sequence is complementary to that of the segment of DNA on which it is transcribed. This has been demonstrated by hybridization experiment in which artificial RNA DNA double strand are produced. Hybridization takes place only if the DNA and RNA strands are complementary. Usually each gene transcribes its own mRNA. Therefore, there are approximately as many types of mRNA molecules are there are genes. There may be 1000 to 10,000 different species of mRNA in a cell. These mRNA types differ only in the sequence of their bases and in length. When one gene, that is cistron, codes for a single mRNA strand, the mRNA is said to be monocistronic. In many cases, however, several adjacent cistron may transcribe an mRNA molecule, which is then said to be polycistronic or polygenic. The mRNA molecule have the certain structural feature. First one is cap. At the 5' end of the mRNA molecule, in most eukaryotic cell and animal viruses, molecules is found a cap. This is blocked methylated structure M7GPP, NMP, MP or M7GPP, NMP, NMP, NP, where N any of 4 nucleotides and NMP is equal to 20 methyl ribose. The rate of protein synthesis depend upon the presence of the cap. Without the cap, mRNA molecules bind very poorly to the ribosomes. Non-coding region. The cap is followed by a region of 10 to 100 nucleotides. This region is rich in adenine and uracil residues and does not translate protein. The initiation codon. The initiation codon is AUG in both prokaryote and eukaryotes. The coding region. The coding region consists of about 1500 nucleotides on the average and translate protein. It is made up of 73 to 93 nucleotides. Each bacterial cell probably contains about 100 or more different types of tRNA. The function of tRNA is to carry amino acids to messenger RNA during protein synthesis. Each amino acid is carried by a specific tRNA. Since 20 amino acids are coded to form proteins, it follows that there must be at least 20 type of tRNA. The next one is transfer RNA or soluble RNA. After ribosomal RNA, the second most common RNA in the cell is transfer RNA. It is also called soluble RNA because it is too small to be precipitated by ultracentrifugation at 1 lakh gram. It constitutes about 10 to 20% of the total RNA of the cell. Transfer RNA is a relatively small RNA having a molecular weight of about 25,000 to 30,000 and the sedimentation coefficient of matured eukaryotic tRNA is 3.8 s. Structure of the transfer RNA. The nucleotide sequence of tRNA was first worked out by Holy et al. 1965 for yeast alanine tRNA. Since then, the sequence of about 75 different tRNAs ranging from bacteria to mammals has been established. The different tRNAs are all minor variants of the same basic type of structure. Several models of the secondary structure of tRNA have been proposed, and of these, the cloverleaf model of Holy is the most widely accepted. Transfer RNA is an essential component of the protein synthesis reaction. There are at least 20 different kinds of tRNA in the cell, and each one serves as the carrier of a specific amino acid to the site of translation. Transfer RNAs are L-shaped molecules. The amino acid is attached to one end and the other end consists of 3 anticodon nucleotides. The anticodon pairs with a codon in messenger RNA, ensuring that the correct amino acid is incorporated into the growing polypeptide chain. The L-shaped tRNA is formed from a single strand RNA molecule that folds into the proper conformation. 4 different regions of double-stranded RNA are formed during the folding process. The 2 ends of the molecule form the acceptor stem region where the amino acid is attached. The anticodon is an exposed single-strand region in the loop at the end end of anticodon arm. The two other stem or loop structures are named after the modified nucleotides that are found in those parts of the molecule. The D arm consists of dihydrouridylyl residues while the TC arm contains a ribothymidylate residue, a pseudourydylate residue, and the sidylate residue in that order. All tRNAs have a similar sequence. The variable arm is variable, just as you would expect. In some tRNAs, it is barely noticeable, while in other it is the longest arm. The tRNAs are usually drawn in the cloverleaf form to emphasize the base pairs in the secondary structure. Unusual bases in tRNA. In addition to the usual bases, adenine, uracil, guanine, and cytosine, transfer RNA contain a number of unusual bases. In this respect differs from mRNA and rRNA. The unusual bases of transfer RNA account for 15 to 20% of the total RNA of the cell. Most of the unusual bases are formed by methylation. Example, cytosine and guanine on methylation yield methyl cytosine and methyl guanine respectively. Precursor of transfer RNA molecules transcribed on the DNA template contains the usual bases. These are then modified to unusual bases. The unusual bases are important because they protect the transfer RNA molecule against degradation by RNAs. This protection is necessary because RNA is found floating freely in the cell. Some of the unusual bases of transfer RNA are methylguanine, dimethylguanine, methyl cytosine, ribothymin, pseudouridine, dihydrouridine, and methyl inosine. In general, organisms high in the evolutionary scale contain more modified bases than lower organisms. Classification of transfer RNA. The differences in the transfer RNA lie in the D arm and the variable arm. Based on the differences in these two variable regions, three classes of transfer RNA have been recognized. Class 1. The Class 1 transfer RNA with 4 base pair in the D stem and 4 to 5 bases in the variable loop. The Class 2 RNA consists of 3 base pair in the D stem and the 4 to 5 bases pair in the variable loop. The third class transfer RNA with the 3 base in the D stem and a large variable arm. A simpler classification based on the variable arm recognize two types of transfer RNA. Class 1 with 4 to 5 bases in the variable loop. Class 2 with a large variable arm of 13 to 21 bases. Tertiary structure of transfer RNA. Electron density map have revealed that transfer RNA has a tertiary structure. The structure is due to hydrogen bonds between the bases and ribose phosphate backbone and between the backbone residues. Initiator of transfer RNA. The starting amino acid in eukaryote protein synthesis is methionine, while in prokaryote it is N-formyl methionine. The tRNA molecule 3 specific for these two amino acids are methionine transfer RNA and N-formyl methionine iRNA respectively. These tRNAs are called initiator transfer RNAs because they initiate protein synthesis. Initiator transfer RNA have certain features which distinguish them from other transfer RNAs, and the initiator transfer RNAs of prokaryotes and eukaryotes also differ. In most prokaryotes, the 5' terminal nucleoside is cytosine. It has opposite it, that is in the 5th position from the 3' end, and a nucleotide. There is no Watson and Crick base pairing between the two. In the blue-green algae, however, the 5th nucleotide from the 3' end is cytosine. In eukaryotes, there is an adenine uracil base pair at the acceptor stem. Specificity of transfer RNA. Two important steps in translation during protein synthesis are the activation of amino acids and the transfer of amino acids to tRNAs. Each amino acid has a specific activating enzyme, tRNA aminosyl synthetase. Thus, there are 20 different tRNA aminosyl synthetases for the 20 common amino acids found in proteins. Some transfer RNA synthetases can activate more than one amino acid, that is, they show only a limited substrate specificity. Thus, isoleucine transfer RNA synthetase can also activate L-valence valine and valine transfer RNA synthesis can also react with 309. The enzymes, however, recognize only a specific set of tRNAs as substrate. L-isoleucine transfer synthetase recognize only tRNA isoleucine, and valine tRNA synthetase recognize only tRNA valine. Thus, specificity is involved at two stages: activation of the amino acid and the transfer of the amino acid to tRNA. Another group of enzymes that transfer RNA, amino acid transferases, catalyze the transfer of an amino acid from the amino acid tRNA complex to specific acceptor molecules. This is all about the structure and various types of RNA. Thus, we concluded this episode. We will discuss a new topic, that is protein synthesis, in the next episode. Thank you.